I'd like to thank ARD for making this possible here today, to dedicate the welcome seminar to highlighting the central role of our biostat and stat departments in fighting COVID-19, and I'll be highlighting today partly through our collaboration with WHO. The topic will be Seeking COVID-19 Vaccines Having Worthwhile Efficacy, and the role of platform trials. And I'd like to acknowledge the WHO Solidarity Trial Expert Group that had a major role in the development of a lot of the ideas that I'm going to be sharing today. So in essence here, I will be focusing on the WHO Solidarity Vaccines Trial, but I do want to maybe set the stage by briefly discussing the enlightenment that we've had from learnings by addressing previous public health emergencies, including the Ebola virus disease outbreak in the Democratic Republic of Congo. So I will start, that's where I will start. And so in essence, as I'm sure most of you, all of you know, we had a major outbreak of Ebola in West Africa in 2014-15. Where sadly, the mortality rate was extremely high. We had about 90% mortality, which led to an urgent need for more effective approaches for both prevention of spread and for treatment. Fortunately, we had then very significant evolution of background optimized standard of care with aggressive IV fluid resuscitation, hemodynamics, other aspects of clinical care, that got mortality down to around 50%. And yet there was considerable interest in experimental interventions, monoclonal antibodies and antiviral drugs. And we had a big debate. Would it be ethical to randomize to these interventions in a public health emergency? And some of us argued that in what setting would there be a more compelling need for reliable insights than in public health emergencies? And randomization is so important to that, especially when there's been such an evolution in what the control arm experience would be. And by the way, as we go forward here, I should mention that ARD will be monitoring the chat feature. So please do enter any thoughts, questions that you have. We would like to very much engage you in this process. So Susan and I published an article that indicated that medical intervention should be evaluated in a manner that would be ethically acceptable as well as efficient and reliable. And yet ethical considerations are not just individual ethics, safeguarding the interests of participants, but collective ethics, enhancing the ability to improve health through timely and reliable insights, which means that efficiency and reliability is part of an ethical process. But critically, we cannot, in the interest of achieving efficiency, compromise reliability. So while efficiency gives us a choice, reliability is critical in our research to obtaining an informed choice. And so fortunately, in the 2 years after the Ebola outbreak in West Africa in 2017 and 2018, we were able to collaborate with the National Academy of Medicine and the WHO R&D and Working Group. And fortunately, both of those highly influential entities came forward with indications that even in public health emergencies, randomized trials are the preferable approach. Unless there are compelling reasons to not do so, every effort should be made to implement randomized trials. And one such randomized trial, important study, was the PREVAIL-2 trial that was being conducted in Ebola. And it had 3 critical features. It was randomized, it used a proper optimized standard of care control arm, and it had a proper 28-day mortality endpoint. And it was designed to start in March of 2015 with 200 participants enrolled from West Africa. The good news is we had a waning of the epidemic. The bad news is that really impacted the ability of the PREVAIL-2 trial to reliably address the questions it was designed to answer. It had been designed for 200 patients. At the waning of the outbreak, there were only 71 patients that were on the trial. And there were only, I think, 21 deaths that had been obtained at that point. And so, in essence, these results then were published. And in the New England Journal, in our article this April, we argued that in light of the ethical principles as described by the Declaration of Helsinki, it was proper that these results were published because this trial was only designed to be carried out during the single outbreak. And we also believed that the wording in the NIH press release that the study finds that Ebola treatment, ZMapp, holds promise, although results are not definitive, was balanced. And yet, in spite of that, given the sense of urgency that exists in public health emergencies, there are real risks in settings like this with overinterpretation and prejudgment about such results. So essentially, if we now look forward here, after the outbreak of Ebola in West Africa in 2016-17, we then had in 2016 to 2018, considerable effort was made in a very proactive way by the WHO R&D working group. And I've already mentioned in part to endorse the importance of randomized trials, but also more broadly to look at establishing principles and infrastructure in advance. Let's not wait for a public health emergency. Let's anticipate when they strike what we want to be, where we want to be so that we can actually conduct effective and reliable research. And we were motivated by many settings, certainly by Ebola, but also by the Zika virus that was very active in South America, by Lassa in Nigeria, chikungunya in West Africa, in the Middle East rather, and Nipah in India. And we were still very worried about coronavirus, having recognized the major challenges it provided to us in SARS in Asia in 2003, and it's the reemergence as MERS in the Middle East in about 2012 to 2014. And so this research that we had done at that time, I think, did position us to be able to more effectively respond when, unfortunately, we had another Ebola outbreak. And this one occurred in mid-2018 in the Democratic Republic of the Congo. And I'll be talking about two aspects of how we responded to this. One, the WHO Compassionate Use Access Murray program and the NIH-sponsored PALM randomized clinical trial. And this very important RCT was certainly enlightened and enhanced by what had been done in a preparation fashion, and yet was building on a scenario where the PREVAIL-2 results had been released for the ZMapp effects, even though those results were really not reliable. So as we then planned to embark on the PALM RCT in mid-2018, there were lots of discussions that we had to address. We had ZMapp, but we wanted to look at other monoclonal antibodies. We had MAB114, we had what we call Regeneron, and we had an antiviral called remdesivir. We wanted to evaluate all of these. Some of us believed that it should be done using an optimized standard of care control arm, while others believed that no, we had PREVAIL-2, we should go ahead and use the ZMapp with standard of care as the control. Those of us that advocated for optimized standard of care said using it in this setting will allow us to actually reliably understand the effect of ZMapp. And by the way, if ZMapp is modestly effective, we get much more insight about a more effective experimental therapy by having optimized standard of care. And if ZMapp is really effective, then if experimental and ZMapp look the same, you won't know reliably whether they're the same, really effective, modestly effective, or ineffective. So therein lied, there was the debate. The study did go forward with the optimized standard of care ZMapp control, so with this design. The results, in essence, from PALM in this really important trial yielded about a 50% mortality on ZMapp and remdesivir, and an absolute reduction of about 15% on MAB114, and about a 20% reduction on Regeneron. So it certainly was a study that provided extremely important insights, and yet it didn't allow us to understand whether ZMapp or remdesivir were effective. It did give us reliable insights about Regeneron, but less reliable insights about MAB114. One other quick comment, one of the other strengths of PALM was that the study started with ZMapp, MAB114, and remdesivir. So it wasn't until during the study that Regeneron was added in a platform trial, which was, as you can see, a great strength of the trial because this was the most successful arm in the study. So Looking then more broadly about what happened in 2018-19 in the Ebola outbreak. The WHO, my heroes in this whole process have been WHO. They developed this compassionate access program called MURRAY. And the concept was participants, people identified with Ebola in Congo that didn't have access to an RCT should at least have access to compassionate access to investigational agents. And they appointed an international oversight committee. I had the privilege of chairing that international oversight committee. And what our committee argued was that anyone that in the Congo that had a diagnosis should get compassionate access. But once the infrastructure was in place to allow participation in RCT, they should no longer be eligible for MURRAY. They should in fact be channeled into the RCT. And so as the outbreak emerged, going from Beni to Batambo to Katwa to Mangina, the sites were remarkable. They carried out exactly that recommendation, using MURRAY during the time that they were setting up infrastructure, but then channeling all of their participants into the RCT. And so even though this was being done, this research was being done in the midst of a public health emergency and in DRC at that time, in the midst of a civil war, where 2 of our sites were burned down as part of that civil war, nevertheless, what the investigators on the ground accomplished was amazing. Of the 3,000 people diagnosed with Ebola in the DRC outbreak, half of them were engaged in either the MURRAY Compassionate Access Program or the PALM RCT program. And this accomplishment was unprecedented, surely in the Ebola domain. So unlike what happened in West Africa, where we got 70 participants randomized into the Prevail 2 trial, 700 were randomized into the PALM RCT. The committee that I was chairing, the MURRAY program, complimented everyone that was involved, saying that the efforts that had been done created a paradigm to develop an infrastructure to deliver compassionate care while efficiently and effectively attaining evidence needed to reliably assess benefits and risks in a public health emergency. And we had a follow-up corresponding publication in the New England Journal about creating a framework for conducting randomized trials in disease outbreaks. And there were several key arguments and advocacy in that New England Journal publication. We said we needed a new approach to clinical trials that would allow for a platform or core protocol concept that would allow a trial when you had a waning outbreak, if those results were not reliable, to extend that trial across multiple successive outbreaks in order to position us to be in a position to prevent prejudgment from early release of results from a waning outbreak. We also argued that new investigational agents should be added to protocols over time in public health emergencies, as was done in the PALM trial that gave us the remdesivir, the Regeneron regimen rather, in a platform trial approach. And that this approach should be in fact implemented in COVID-19. And this is happening. I'll be talking for much of the presentation today about the WHO Solidarity Vaccines trial, but I want to briefly mention that WHO also has a Solidarity Therapeutics platform trial, and the UK had a very important recovery trial. And these trials were instrumental here thus far already in COVID-19 in looking at how we can reduce the risk of mortality. The ultimate interest here is reducing the risk of mortality in hospitalized patients. And the results often weren't as expected. Dexamethasone steroids was very effective, particularly in the most advanced participants. And some other regimens that had been claimed to be very effective, hydroxychloroquine, Kaletra, lopinavir, ritonavir, were not effective, were found to be ineffective in this trial. There are other important efforts being made in pre-exposure prophylaxis in healthcare workers, and post-exposure prophylaxis in nursing homes and household contacts. And in early disease settings, there are important studies that are ongoing. And a key comment that I want to make here is that the departments of statistics and biostatistics here at UW and the Hutchinson Center have been instrumental. Many faculty in green here have played very significant roles, and there are many others as well from Seattle in biostat and statistics. Yunda and Holly and Peter and Bets and Gary and Alex and Elizabeth have all been very significant players. I want to go on and talk a bit about the Solidarity vaccine trial, but maybe pause here for a moment to see, Yardi, if there are any questions to this point.
No questions yet, but maybe we can give 10 seconds. People can either type the question in the chat box or ask us to unmute them and pose the question themselves. Yeah, no questions, so feel free to go
Okay,
ahead.
so let's press ahead to actually now talking about the WHO Solidarity trial for COVID-19. And I'm excited to be able to indicate that we have every expectation of launching this key trial sometime later this month. We've recently published a Lancet article that I think has been shared with all of you by ARD. That was developed by our WHO Solidarity expert group. And this group identified 3 critical issues in planning COVID-19 vaccine trials. First of all, we argued that it's important that we demand not only proof of some vaccine efficacy, but proof of worthwhile vaccine efficacy. And the idea behind this is that a weekly effective vaccine can do more harm than good in a COVID-19 pandemic. In essence, a weekly effective vaccine could reduce social distancing if vaccinated individuals wrongly believe that they're immune. We often call this disinhibition. If I think I have some protection, I might feel more entitled to re-engage in more complete interactions, and that could have a net negative effect. Or it could reduce policies by authorities for social distancing or mask wearing or other procedures if the authorities wrongly assume that the vaccine provides substantial risk reduction. It's also problematic because a weekly effective vaccine could interfere with the development and evaluation of other better vaccines. And the problem there is subsequent vaccines may need to be compared to that weekly effective vaccine in non-inferiority trial designs. And while they give interpretable results, they often require several-fold more person years of follow-up than a placebo-controlled trial, and they have risks of interpretation with issues such as biocreep. Also, though, really importantly, a weekly effective vaccine could adversely affect public confidence in vaccines. We know in COVID-19 that if we're going to be able to achieve herd immunity, it's not only going to take an effective vaccine, it's going to take wide uptake of that vaccine. And that's true for other diseases as well. And if our first generation vaccines turn out to be recognized to be much less effective than they're touted to have been initially, that confidence when you have an effective vaccine may no longer be there. So this is really a critical issue. There are 2 other really critical issues that we identified in this article. And the first of these is whether there should be significant focus not only on single vaccine trials, but on working together to do a more efficient multiple vaccine platform trial. And also to assess not only what the effects are on preventing any kind of infections that have symptomatic disease, but protection in particular against severe disease and understanding the duration of protection and having long-term safety and risk of assessment assessed or evaluated, risk of enhancement rather, where enhancement is a real worry with vaccines, and certainly with vaccines in the coronavirus domain from our experiences in SARS and MERS, that vaccines could actually enhance or worsen the overall risk of disease and important disease. And so as we'll talk later on, it's important then to have longer term follow-up to be able to address these issues reliably. So in July of this year, the WHO sponsored a very important forum that was the COVID-19 Global Research and Innovation Forum. We had more than 1,000 participants in that forum. And one of the key leaders of that meeting, Marco Cavalieri of the European Medicines Agency, noted that we had an impressive number of almost 1,200 registered therapeutics randomized clinical trials. Yet to quote him, what he observed in July is that very few of these studies had provided the evidence necessary to meaningfully inform clinical practice. And one of the other participants at this meeting, Mike Jacobs, who was a member of that committee that I was chairing, the international committee that was overseeing the WHO Murray program for EVD and DRC, acknowledged that the remarkable power of large platform trials is critical, and there's a need for global collaboration on such trials if we want to achieve timely and reliable assessments of vaccines. And so, fortunately, this collaboration is happening. So what I'd like to do in much of the rest of the presentation here is briefly present what is, in essence, the WHO Solidarity Platform Trial that's been developed through extensive collaborations by the scientific community, including significant leadership from biostatistics and from statistics. So as he already mentioned, I'm a member of the steering committee. The steering committee has had 5 core members who've been working intensively around the clock for the last 3 to 5 months in preparing for where we are on this platform trial. And interestingly, 3 of those 5 members are statisticians. We've had terrific leadership from Anna Maria Henua Restrepo, who is clinical lead from WHO, and from Phil Kraus, who provides significant clinical and regulatory leadership from FDA. But the other 3 members are biostatisticians: Ira Longini from University of Florida in Geneva, Richard Pito from Oxford, and myself. So biostatistics has certainly, and statistics has had a major role here. Our goal has been prompt, efficient, and reliable evaluation of efficacy and safety of several candidate SARS-CoV-2 vaccines. And so this slide, in essence, attempts to lay out several key concepts and principles in the design and conduct of the WHO Solidarity vaccine trials. So you're seeing across the top row here in blue concepts, and then across the bottom row here are key consequences. So our concepts here are, first, to be evaluating several different candidate vaccines. Second, to expeditiously enroll participants at sites that have high rates of COVID-19. Thirdly, to eliminate inefficiency of designing and conducting separate trials. And finally, to benefit from the international collaboration. And so in essence, you see the arrows going down of these various steps that lead to the consequences. And one of the key consequences is increasing the likelihood of finding several effective vaccines. And we have a priori criteria for selection of these. There are about 120 candidate vaccines that are emerging, where we have adaptiveness, flexibility. We're going to permit the vaccines to come into this trial whenever they're ready. And this and other adaptive features, we hope, will aid our ability to get rapid accumulation of data to support a rigorous evaluation. We're going to have a mixture of fixed sites and pop-up sites. I'll talk about this in the next slide. We'll talk about these pop-up sites as being mobile sites to help chase the pandemic where it is, as part of our approach then to be able to achieve results within 3 to 6 months after each vaccine is ready for inclusion. Part of the efficiency to do this is going to come from having a shared placebo at any point in time for the K vaccines that we're going to be assessing. And when we reach points where we now have established effective vaccines, when placebo can no longer be used, we will use that effective vaccine as an active comparator in a non-inferiority trial. The press has been very astutely following this. We've had many contacts from the press, just hearing in the last couple of days from Carl Zimmer from the New York Times, who wants to understand non-inferiority. How are we going to do this non-inferiority assessment? So we'll be talking to him, I think, at 5:30 on Monday morning, as we try to help them understand some of these critical issues that we're going to be facing in the design and conduct of the trial, where ultimately we hope to achieve an international collaboration that we hope will foster international deployment with equity of access. This is really key. We are not going to overcome this pandemic by getting effective vaccines in the US and in Western Europe. That's not how we're going to stop a pandemic worldwide. We need to be focusing, as WHO is trying to do, on international deployment. So a little bit about the design for Solidarity. It is an international, multi-vaccine, adaptive, shared placebo, event-driven, individually randomized clinical trial. And so if we have K vaccines, and by the way, this is dynamic, the number of vaccines we're studying at any time can be changed over time. It's a time-dependent factor. But at any time, if you have K vaccines, we randomize people 1 to 1 to 1 to 1 to 1 to those K vaccines or to the control arm. In essence, we will have additional features that are going to be critical to the design. During this morning's pre-dawn meeting that we had, virtual meeting that we typically have often each week, where this meeting was between our WHO steering committee, and this morning we had more than 100 of our prospective fixed sites that were represented. I was strongly encouraging all of them to be engaged, to continue to be engaged, and that we're going to be bringing them in waves, where they're going to be brought in according to the nature of their current epidemiological data for the nature of viral burden in those settings. And so those will be our fixed sites. But we'll also have mobile sites in vans where we'll chase where the pandemic is, where the viral load is, to allow us to achieve the greatest likelihood of substantial numbers of events that will increase the power. And these procedures will be used to increase the rapidity of results while maintaining reliability and hopefully be able to help us in identifying multiple successful vaccines. So the primary endpoint will be rate of virologically confirmed, PCR confirmed, symptomatic COVID disease, regardless of symptoms. So if we achieve, as we're hoping, the enrollment of more than 20,000 participants per arm in less than 3 months, and we do so with high risk, where these participants have an attack rate of 1 to 2% over 6 months, the good news is we're going to achieve insights about the rate of the effects of these vaccines on the primary endpoint of virologically confirmed COVID-19 symptomatic disease within about 3 months of the time that each participant will have finished their final dose. So they're going to be on 1 to 2 dose vaccines. And so within a relatively short period of follow-up, we'll have this answer. The bad news is that won't be telling us about duration of efficacy. It won't be telling us about whether or not there could be waning immunities that could lead to an early effective vaccine ultimately having longer-term risk of enhancement. And it won't be telling us reliably about whether we're affecting severe disease and fatal disease. I think we think from vaccine experience that when you have vaccines effects against any severity, that you're likely to be effective against what we care about most, which is severe or fatal disease. But that won't be known because when you have 150 cases of any severity, we'll only have about 25 to 30 cases at most of fatal events. And so a critical feature that we're proposing in this trial design is that we will continue follow-up. If we find a vaccine that is effective in reducing the occurrence of disease of any severity, we will continue to follow those participants on their randomized arm. We'll release the data to regulatory authorities to begin the regulatory process for ultimate EUAs or dissemination of those vaccines. But there's also manufacturing and distribution, particularly on a worldwide basis, that could take considerable time. And so in that time period, by following the vaccinated and placebo groups, we will get far more important insights about the overall true benefits and risks of these vaccines. So there will be also, at some sites, blood samples taken at baseline and post-vaccination and longer term, because we also, as a secondary endpoint, would like to know whether or not the vaccine increases or decreases the rate of asymptomatic infections, because this is critical or very important to the effect of a vaccine in its potential to induce herd immunity. We will also be looking at immune responses and immunologic biomarkers to understand correlates of risks and correlates of protection, which takes multiple studies over time to adequately understand. Vaccine safety will be key. And so one of the principal objectives will be to look at SAEs over time and adverse events of special interest. Will also be, importantly, as we've mentioned here, having long-term follow-up for safety monitoring that will allow us to assess the possibility of enhancement, unfortunately inducing enhanced incidence or severity of disease over the longer term. For reasons I'll talk about, it's important the study be double-blinded, although that's easier said than done. Vaccines can have differing schedules. And so we might be looking at multiple vaccines, A, B, C, D, against a placebo. And we might be looking at any time about one vaccine against control, or 2 or 3 or 4. So suppose we have at some point in time 3 experimental vaccines, A, B, and C, against placebo. It has to be blinded. So do we have to give the A's both B and C placebos, and the B's get A and C placebos, and the C's get A and B placebos, and the placebos get A, B, and C placebo? That would be an overwhelming complexity with all those placebos. So we're using I think Jim Neaton developed concept from around the early 1990s in HIV research, where we actually will do a 2-stage randomization. We will, in an unblinded way, randomize you to the A group, B group, or C group. So you'll know your color. But within each group, in the 4 patients on A, 3 in a blinded way will get active A, 1 in a blinded way will get placebo. Within the B group, 3 will get active B, 1 will get the placebo. And within the C group, 3 will get active C, and 1 will get the placebo. So it's an intriguing approach to achieve a blind with only needing to provide placebos to a very small number of participants. So additionally, what's very important is that the study is going to be involving active rather than just passive follow-up. So we will be doing weekly automated follow-up. So we will be contacting the participants every week, asking them whether or not they have, per WHO case definitions, relevant COVID-19 symptoms. And if they do, that will then trigger PCR virologic confirmation of whether they actually have SARS-CoV-2 infection. So this makes it critically important that we have a blind. We don't want people reporting their symptoms differentially based on the knowledge that they're on the control arm versus an active vaccine. And we'll also then be, in each vaccine, we'll be monitoring over time for early evidence of benefit or lack of benefit to allow the study design to be more efficient. So we'll be using O'Brien-Fleming monitoring boundaries. So we'll have early termination for a beneficial vaccine if the effect is so strong that even adjusted for multiple testing, you can rule out 30% or less vaccine efficacy. Or for lack of benefit, if the early results are so unfavorable that adjusting for multiplicity, you rule out that the true efficacy is at least 60% or more. The sample size, the trial will be event-driven. And so the main analysis will be triggered by the occurrence of a total of 150 cases of primary endpoints, which will be virologically confirmed symptomatic COVID-19 across the 2 treatment arms. And so, in essence, a successful candidate vaccine will be an estimate of at least 50% protection, risk reduction, where if you have an estimate of 50% risk reduction with 150 events, that gives you precision to conclude that true vaccine efficacy is at least 30%. And so with the O'Brien-Fleming monitoring boundary, as my steering committee colleague Richard Pito often says, the way this is going to work is when you have 50 events, you stop early if the imbalance on events between placebo and active vaccine is at least 40 versus 10. When you have 100 events, if it's at least 70 versus 30. When you have 150 events, if it's at least 150 versus 100 versus 50. So in essence, compared to doing separate trials, this multi-center, multi-vaccine, shared placebo platform trial will, we hope, provide timely and reliable evaluation, and engaging 60 countries and 250 sites will allow us to have timely acquisition by rapidly enrolling a large number of participants at high risk. So in essence, conclusions regarding the platform trial is that our goal, our hope is that this will allow us to proceed efficiently, to obtain reliable evidence, to support decision-making about global vaccine deployment of multiple safe and effective vaccines with an equity of access internationally. There's one more section here, Yardi, but let me just pause to see if there's any questions to this point.
So yeah, we have a couple of questions. And the first one reads, will vaccines that have already started in phase 3 trials also be included in the Solidarity trial?
So good question. Our expectation is that there will be a mix of vaccines that are done as single vaccine trials. There are a number of these that are ongoing. Moderna and AstraZeneca, Pfizer and others have such studies underway, and they will proceed independently of WHO. WHO is screening large numbers of potential vaccines that have preliminary phase 1 and phase 2 data that are in humans that are looking at whether or not they're generating the intended immune responses. But those vaccines that would be entered then into the Solidarity trial would be vaccines that hadn't yet been studied yet in phase 3.
Then we have another question that says, well, thank you for this excellent presentation. Could you comment on the reasons why the WHO vaccine trial has chosen a placebo arm rather than an active comparator, such as another unrelated vaccine?
Yes, so either approach, I think, has its merits. There have been a number of settings where people have used other unrelated vaccines that are anticipated to not have an effect on the outcome. And in this case, what would be critical if we did so would be to ensure that they wouldn't impact other infectious processes that would induce symptoms that would be similar to COVID. So it was the sense that the purest approach here would be to randomize people to standard of care, which is what the vaccine is. You're getting the vaccine, placebo vaccine, in addition to standard of care against the experimental vaccines. And it's my sense that I think that's how most are proceeding right now with COVID-19 vaccines.
Okay, we have a third question, which is, what vaccines are lined up to be in the Solidarity trial, and what are the first wave of vaccines for this trial?
So that's an issue there are many subcommittees that are being set up by WHO. We have a subcommittee that is involved in the vaccine selection process. They've been intensively meeting on a regular basis. So we are at this point blinded to that. We don't yet know, but I was hearing this morning that it could be within the next couple of days that we'll be informed about what the first wave of selected vaccines will be.
Thanks, Tom. We have one more question. Well, two really. So do you need to update the sample size calculations based on when new vaccines have been added and when the trial switches to a non-inferiority trial?
Great question. Yes, in a major way. So I'll come to this research in a minute. In fact, it motivates this last topic of future research. So one of the topics I want to mention that I'll come back to is we're looking right now into what would be, from a research perspective, the preferred approaches to study new vaccines once we have an established vaccine. So I'll come back to that thought in a moment, Iardi.
Yeah, maybe a final one. So a general question, if the study are terminated early given the early evidence of efficacy, how will the follow-up be done to ensure longer-term safety? Will there be any changes?
Yeah, that's a critically important point. So what I noted earlier is if we're successful in enrolling the large number of participants at high risk as we're planning, we will get a relatively early answer about the effect on whether symptomatic disease is reduced at any level of severity. But we will not have insights about long-term. And so it's our plan to continue to follow people in a blinded way on their randomized intervention until such time as there's deployment of an effective vaccine at their site. And that will aid us hugely in being able to get necessary long-term insights.
Okay, then maybe one more, Tom, and a final one. So it says, thanks for the presentation. Any thoughts on doing testing for asymptomatic disease for the primary endpoint? I imagine it's very costly, but I've read that even with asymptomatic disease, some subjects have things like cardiac damage.
Yeah, that's an important question. There's been a lot of discussion about this. I think there is fairly uniform approaches that we are focusing on symptomatic disease. There is a sense, it's not known, but there's a sense that we will have increasing efficacy by increasing severity. So we may have the biggest challenge with a vaccine in reducing asymptomatic disease. We'll have more success in preventing symptomatic disease, and potentially the greatest success, which is really important, in preventing the most severe disease. And that is ultimately a critically important objective of a vaccine, is to prevent hospital disease infections that are so severe that it leads to hospitalization and death. And the worry is if we include all infection, including asymptomatic, that could greatly dilute our sensitivity. So that's going to be assessed as a secondary endpoint. And it's more complex to assess because it'll take blood draws at baseline and in follow-up to be able to assess the effects on asymptomatic. So the last topic here is that just to really highlight, there is a need for statistical contributions to the COVID-19 research efforts. And obviously, as we've talked about, this includes in the design and conduct of trials, but it also needs to include publishing in journals that are clinical trials methods or appropriate methods in frontline journals. And I've already mentioned two such publications, the Lancet publication about the need for worthwhile efficacy, and the New England Journal publication about the need for platform trials. But there have been a number of others that our group's been involved in. Just to mention a few, it's important to be able to protect the integrity of non-COVID trials during the pandemic. And so we've talked in May of 2020 about approaches to achieve adherence and retention in the COVID setting that would be comparable to what you would have in a real world setting without COVID. We've talked in July about the importance of optimizing DMC, Data Monitoring Committee functions, since these committees during pandemics could be meeting 10 times as frequently as they would typically be meeting. It's important to be efficient with factorial designs. We published an article recently about how factorial designs could be important in HIV prevention. And these ideas are important also in COVID, where if you have a hospitalized patient with severe COVID-19 disease, they should be receiving a steroid such as dexamethasone, but we could be randomizing them to multiple factors, to antivirals, yes, no, or to agents for cytokine storm as more efficient ways of studying effects in COVID-19. And clinical endpoints are important. We care about immunogenicity measures, immune response measures, but ultimately we need direct measures of how we are affecting how a patient feels, function, and survive. So can we prevent hospitalizations or deaths? Can we prevent time in the hospital or reduce time in the hospital? Or for an uninfected person, can we reduce their risk of infection? And as I mentioned, we're working now on non-inferiority methods to get at the question that you were asking earlier about when you have an established vaccine as the control, how do we do this in a we look at new vaccines against the established vaccine in a manner that they're non-inferior. And that methodology exists, but how should it be amended to be applied to a setting as in COVID-19 where our goal is to get many safe and effective vaccines? Or if you've started a trial for a new vaccine against a placebo and now it switches to an established vaccine, how do we proceed in that manner to allow the aggregation of the data in the hybrid design? So these are methods that are currently underway in development. So it's clear that we need statisticians. We need statistical input here in the front lines, not only in the design of and the conduct of studies in public health emergencies, but in the development of appropriate methodology. So my last slide is that it brings me back, interestingly, to the 6th Seattle Symposium. And the reason that it does is that, as Laura just pointed out, this is an important time in our School of Public Health. We're starting this year, right now, the celebration of our 50th anniversary, coinciding with our campaign for students. When we had our 25th anniversary, Gil Oman challenged us to think of creative ways to highlight the importance of what we do at the School of Public Health. So we started the Seattle Symposium in Biostatistics, where we have one every year. Well, now we are at our 50th anniversary to the 6th Seattle Symposium. And we've had remarkable leadership in this by Marco Caroni and Noah Simon, who are our chairs, who put together an amazing agenda for what we will call, or what is called, precision health in the age of data science. Go to the link. The link is giving you impressive details about the symposium. And I'll just highlight a couple of thoughts about this. The symposium itself is Saturday, Sunday, Monday, and Tuesday mornings on November 21st to 24th. On the 21st, Lisa McShane is the keynote speaker on precision diagnostics. She's been a world leader. She's at NCI, and she's been a world leader in development of such the methodology for such diagnostics. On Sunday morning, on November 22nd, Rob Califf, who's the former commissioner of FDA, will be talking about clinical trials with targeted therapies. On Monday morning, the 23rd, Scott Ziegler will be talking about learning from observational data. And then on Tuesday, we will talk about data science in the age of COVID-19. So we've gotten into these issues here in 45 minutes today, but we'll have 4 hours. And I'm delighted that Phil Krause is going to deliver the keynote. And if you look at the top, Phil is the first author of the Lancet article we shared with you on worthwhile efficacy. And one of our key presenters on November 24th will be Natalie Dean, who is the first author in the New England Journal publication. So one other quick thought about this is that the overall fee structure is one of the real positives that come out of a virtual format. When we held the 2015 symposium, faculty members who participated paid $375 for the symposium and $975 to attend all of the short courses. It's $150 now in contrast. Students paid $100 and now it's $30. And they weren't in fact even given a short course fee and an option for that, and it's $30. So it's a remarkable opportunity to engage in this process. We encourage all of you to think about attending. And I guess my final thoughts here are that much has been achieved to date in public health research, and yet so much more has to be accomplished in the treatment and prevention of diseases overall, and certainly in public health emergencies. So we hope that sessions such as the 6th Seattle Symposium and courses, and I'll pitch Biostat 524, are places to actually help advance the knowledge that we have that will be so critical. I say in particular to our biostat and our statistics students that my sense is you've chosen great academic fields if your interest is in making a difference. We need you. You can make a significant difference in health. And we'll thank you in advance because you're our future for doing this. So Ayardi, back to you. Thank you.
Thank you, Tom, for the informative and inspiring talk. So we have a question from Marco, actually. He's asking, what are the implications on the proposed study in design and statistical analysis if an emergency use authorization is given to a vaccine candidate before the end of any phase 3 trial?
Yeah, great question. There's been a lot of discussion, and it's part of the motivation for our Lancet article. We will hope that there will be proper, measured, scientific, and regulatory decision-making about the reliability of evidence. We all want to have effective strategies to address the spread of COVID-19, but the goal isn't to have a choice, it's to have an informed choice. The goal is to ensure that we maintain regulatory standards. I know there's a letter being circulated right now to be sent to leadership, administration leadership, to say it's critically important that science is directing how we proceed in dissemination of vaccines. EUAs may in fact be an appropriate strategy to go forward. We will be making decisions about when it's necessary, when there's sufficient evidence about the benefit to risk of a vaccine to declare from ethical and scientific principles that it should become the active comparator. But one of the real significant issues here is this is an international pandemic. We might have a vaccine that could in fact be disseminated in part of the US or in part of Western Europe. Yet we have a need for many effective vaccines. This is not like many other settings where one effective therapeutic could be developed in sufficient demand to cover world need. We're going to need many effective vaccines. And even if we have a 75% effective vaccine that might be used in the US, we need to have vaccines for Asia and for Africa and for Eastern Europe and other sectors of the country that, in fact, if they are 50% protective, will be major advances on a more global basis. So these are very important scientific and ethical issues that we will have to address. There will be a time, we're hoping, where we have clear evidence of effective vaccines with sufficient supply of those that what would be relevant in any setting would be to understand a new vaccine against that active comparator, in which case then we will go to non-inferiority trials.
So next question is on whether there are concerns on the virus mutating rapidly and outtracing our vaccines.
Yes, and obviously we've lived with this with HIV. I had the privilege of working in the early days with our HIVnet, HIV network, going back to around 1993. And it was our thought at the time, or was our goal, that we would have a vaccine within a few years. And we are now 27 years down the road. We've made major advances in understanding vaccines in that setting, and major advances in being able to use antiretrovirals to suppress viral load, to have radical positive effects on transmission when you're either treating the infected or in a PrEP or PEP setting where you're treating the uninfected. So yes, we know that that feature could add to challenges of effective vaccines. It does mean that we will need to understand what is the effect over extended periods of time. And it does mean that we're likely to need multiple effective vaccines until we can get through those vaccines and other approaches an ability to achieve a level of herd immunity that we can then move forward without as much dependence.
Perfect. One more question. Thank you so much for the informative talk. With production of vaccines before the completion of phase 3 trials, I wondered if there are concerns about wide circulation and use of vaccines before impacts of vaccines on disease severity, especially enhancement, are understood.
Yeah, it's a really, we worry about that. That's an important question. So we're trying to have our cake and eat it too. I mean, the approach that we're trying to take in as creative a way as we can is to identify a proper primary endpoint, which would be virologically confirmed symptomatic disease. And as soon as we know we have that, disseminate that result to the sponsors and to regulatory authorities for them to make proper decisions about dissemination or implementation of that vaccine. But that will take time. And the manufacturing process will take time. In the US alone, we need more than 100 million doses of 2-dose vaccines, hundreds of millions of doses. And so getting effective vaccines that show promise on reducing by at least 50%, by estimate, at least 50%, the risk of transmission to symptomatic virologically confirmed disease, we will continue to follow people and we will get, we hope, significant enhanced insight about durability of effects and about effects on severe disease and risk of enhancement. So that by the time we would actually implement those vaccines, when it would no longer be appropriate to be depriving those randomized participants of that vaccine, we would hope to know much more than just what was its shorter term effect on preventing symptomatic disease.
We have another question. Any predictions or guesses on when really effective vaccines will be available?
I'm dodging the question by saying, hopefully when we have adequate scientific justification for their availability. That didn't answer the question as to when that will be. But we do hope that the international effort will be enabling us to do so in the shortest time possible. We need the world. We need an entire aggregated world effort. This isn't something we do in the US alone, or in Western Europe alone, or any other place alone. We do it together, and we'll do it with a combination of trials that are studying individual vaccines with, I hope, a widespread or widely engaged, efficient approach of a platform study that will look at multiple vaccines. But by working together, I know there are over 100 candidate vaccines. Many of them won't be judged to be worthy of being studied in phase 3 trials, but many will. And hopefully by engaging the entire world, and WHO is engaging more than 60 countries, 70 countries in 250 sites, that complementary to what else is going on by other single vaccine studies, we will together be able to get timely and reliable insights about many vaccines, hopefully a number of which would be reliably shown to be safe and effective.
Hi, Tom. This is Tim Thornton. Quick question about the different rates of coronavirus affecting African Americans and Hispanics compared to white Americans. And so I just wanted to get your thoughts on what are some of the reasons you think might be causing that, as well as is there implications for different vaccines that may be more effective based upon race or ethnicity?
Yeah, it's a great question, Tim. And we hope to be able to gain important insights about that. As you know, it will be particularly challenging to reliably understand that as subgroups. And we care a lot about what you're saying as it relates not only to effective vaccines, but effective therapeutics, that we need to be able, particularly for those populations that are bearing, unfortunately, a larger fraction of the burden of this, our most timely and reliable insights about how to effectively treat, how to prevent infection, how to address severe disease and hospitalize patients in the most effective way. And so a lot is unknown, Tim, about that at this point, and yet there's a lot of intention for us. Richard Pito talks about this all the time, but he wants really robust evidence. He wants us not just to understand in a marginal way whether we have effects in the aggregate data. He wants us to be able to have sufficiently rich insight that we can understand what the influence is about factors, risk factors that you've mentioned, as well as age and other factors. And we'll do our best to address that, but it's going to be one of the significant challenges to get adequate data to do that.
Great. Thank you, Tom.